Hi everyone, my name is Taylor Vickers and today I'm going to be giving an overview of the shared research project I was a part of in Genetics 312 about DNA sequencing and sourdough starters. Sourdough is a type of bread that originates from yeast and bacteria. A basic sourdough starter contains flour and water. In terms of microbes, sourdough is known to contain lactic acid bacteria, more specifically lactobacillus and yeast. Sourdough has the ability to rise due to the production of gas, more specifically carbon dioxide from yeast, which serves as a leavening agent. This shared sourdough project worked to answer these overall questions. How does the type of food provided to the microbes affect the sourdough starters? How does it affect what kinds of microbes will grow? During the first half of the semester at home, the class cared for starters containing flour and water, in addition to some starters containing flour, water, and a variety of fruit. The starters were fed using a feeding schedule, observing rising rates, and even documenting smell in a lab notebook. Afterwards, starter samples were collected from students in which the DNA was extracted for DNA sequencing. The results were then shared with the class using the class data as a whole to answer research questions. My specific experiment used 3 starters: a control starter, which consisted of flour and water, a fruit starter, which consisted of flour, water, and banana, and another fruit starter, which consisted of flour, water, and blueberry. All of my fruit were freshly grown and the flour was obtained locally. I began using a 24-hour feeding schedule with dechlorinated water, placing my starters away from direct sunlight and looking for the characteristic hooch layer to form. Over the duration of caring for my starters, I developed the following hypothesis. The starters with fruit would be different in texture compared to the control starter due to the chemical makeup of fruit making them more watery. This hypothesis was eventually supported, in which I had to switch to a 12-hour feeding schedule to limit runniness in my fruit starters. My starters exhibited phenotypic changes to show that they were feeding and growing, even exhibiting air holes at some points. While my banana and control starter showed the most change overall, one common characteristic of my 3 starters was that they all had a strong, unpleasant smell. Overall, my experiment was a success with 2 of my starters, the control and banana, showing rising rates approaching 2 centimeters. However, each starter exhibited rising and falling at different times. A rising rate experiment conducted with another member of the class shows the control having the greatest increase in growth in centimeters over time. After documenting the physiological changes in my starters over a set number of weeks using my blog, the samples were then sent to undergo DNA sequencing to help determine the microorganisms in the sample by the order of base pairs in the nucleic acid sequence. Only a few genes or regions were sequenced within the genome, which were the 16S rRNA gene and the ITS region. This is called amplicon-based metagenomic sequencing. 16S RNA genes encode for the RNA portion of the 16S subunit of the ribosome, while the ITS, or internal transcribed spacer region, serves as a spacer and varies among fungal species. Using the data from DNA sequencing, I decided to ask the following question. How does the microbial composition of starters containing fruit, blueberry, compare to the microbial composition of the control? My hypothesis was, while the blueberry and control starters may share some similarities, they will differ in overall microbial composition. My hypothesis was supported that there was a difference between the blueberry and control starters, however, it was not to the extent that I originally believed. Here is a look at the Level 7 16S sequencing data comparing the control and blueberry samples. The blueberry samples 15 and 16 contain mainly firmicutes and lactobacillus. Sample 16 also contained a moderate frequency of proteobacteria. The same can be said for control samples, which also contain firmicutes and proteobacteria. However, one difference is that some control samples contain cyanobacteria. These microbes were not particularly surprising since my preliminary research showed that they are commonly found in sourdough. Here's a look at the Level 7 ITS sequencing data comparing control and blueberry samples. A large majority of the samples contain fungi Ascomycota in addition to two other kinds of fungi. The blueberry samples 15 and 16 were similar to one another. However, sample 16 contained a larger frequency of Ascomycota. One difference between the control and fungal species was an increased presence of Diathyrodomycetes in the control samples. After completing this project, I'm curious to see if the pH of fruits affects microbial composition. This is something that I would potentially like to look into. Blueberries are considered to be a more acidic fruit, therefore it would be interesting to see if there is a difference in composition when looking at samples containing more basic fruits, such as bananas, melons, and peaches.